Good afternoon. Thank you to the organizers for the invitation to speak here today. My name is Ryan Demmer, and I'm going to be speaking on the topic of microbiome and cardiometabolic disease, and hopefully trying to say a few things about the relevance potentially to the urban health context and things we can do in the green space to improve microbiome metrics and potentially influence cardiometabolic outcomes. At the outset, I always like to remind people that cardiovascular disease is important. I think everybody knows that, but as was mentioned this morning in some of the sessions, we've become a bit numb to it because it's so highly prevalent it seems almost normative. And it's compounded a bit due to the fact that over the last 50 to 60 years, we've actually made remarkable progress in terms of cardiovascular disease. Nevertheless, despite nearly 50 years of declining cardiovascular disease rates, there are a few important truths. It remains the leading cause of death globally. Cancer, very recently, yesterday I heard, is maybe displacing cardiovascular disease as leading cause of death in a couple of developed countries. But generally speaking, CVD remains the leading cause of death around the world. Very importantly, the absolute burden of cardiovascular disease is rising almost every place in the globe, largely driven by an aging population. And more recently, there's pretty good evidence that rates of CVD are starting to level off. The declining rates of CVD are starting to level off in younger individuals and maybe even starting to increase. And that's very concerning, as it suggests that there are maybe some new risk factors we need to be aware of, or context has changed in a manner such that traditional risk factors are less important. And of course, we know that diabetes and prediabetes rates have been going up fairly steadily for some time. And this, of course, then feeds back onto the larger cardiovascular disease problem and may actually explain some of these leveling or increasing CVD rates in younger age groups. So these are important things to be aware of. And a question I've been asking for many years is what is the potential for microbial etiologies of chronic and particularly cardiometabolic diseases, and what are novel risk factors for these outcomes? And very recently, the microbiome has emerged as one possible novel risk factor of interest. So I want to spend a little bit of time talking about what the microbiome actually is and giving some proof of principle examples as to why the microbes in and on our body space might actually influence chronic diseases and cardiovascular disease in particular. So the human microbiome is often defined as the collection of microbes living in and on our body space. It's been estimated early on that the number of microbes in our body outnumbers our own human cells by an order of magnitude. That estimate has been updated. It seems to be closer to 1-to-1, but nevertheless, if you think about it, it's really crazy to think about the fact that at the cellular level, we're more microbial than we are human. And that just blows my mind every time I think about it. More important than the actual microbes that make up our body are the genes that they carry. And the genes do outnumber our own genome by several orders of magnitude. The microbial genome is roughly 2 million. Our human genome is roughly 23,000. And just as microbes can clean up an oil spill, they can do very important things on behalf of their hosts, such as digest food and train the immune system. So when we're thinking about microbiome, we're really very interested quite often in the genes that they carry and the things that they do for us. So let's move into a couple of proof of principle examples as to why microbiome could be important. The first example is related to gut bacteria and dietary energy harvest. This is probably something most people have heard a little bit about at some point in time over the last 10 or 15 years, and I love this example because it's fairly provocative, and it's really one of the first examples of good science coming out in the microbiome space. And I should say and remind people that microbiome is, of course, incredibly new. The term was coined by Letterberg in 2001, so it's truly a 21st century health topic. In terms of microbiome, the first study I often like to talk about is an animal model. And what they did was to compare energy expenditure and body composition between germ-free mice. These are mice raised in a facility, so they have no microbes, no microbiome. And they compared them to conventionally raised mice with normal gut flora. And what they found was fairly profound and has been repeated in other studies since, was that after only 14 days, germ-free mice accumulated almost 30% more body fat. And that happened despite the fact that they ate more and exercised less. which is a little bit crazy if you think about it. And the implication in these highly controlled animal models is that somehow microbes are extracting food, certain microbes are extracting energy from diet and placing it into fat more efficiently than other microbes. And if you have a gut microbiota tilted towards those microbes, it could be a reason that you more efficiently store fat. And they went on to show that this trait is actually transmissible, at least in mice. They found that if they gave germ-free mice a fecal transplantation from a genetically obese mouse, that mouse would become obese, and the only explanation for this was the transplantation, and they had control groups where they did fecal transplantations from lean mice, and the lean mice recipients did not go on to become obese. So this demonstration of transmissibility really, I think, helps us think a little bit differently about how chronic diseases may spread. It's very unlikely that obesity is a transmissible phenotype like that in humans. That idea is not very popular, but it could be transmissible in terms of the health behaviors that drive microbiome or the environmental factors that drive microbiome, which will become more relevant as we get to the second half of this talk. Another really interesting and recent example that was just published in the spring deals with the idea of performance-enhancing gut microbes. The basic premise is that when you exercise, of course, you produce lactate, which goes to the liver, and some of it goes back to glucose, but some of that lactate actually ends up in the gut, and it enriches a certain subset of organisms that use lactate as a carbon source. When they metabolize that lactate for their energy source, as a byproduct, they produce short-chain fatty acids, namely propionate, which then can go back into muscle and seems to help enhance exercise performance. So you can run farther and longer with less physical exertion. And you can see how this feedback loop ensues. So to test this hypothesis recently, some researchers at MIT and Harvard found some Boston Marathon participants, and they looked at them both before and after running the marathon. They found sedentary controls to compare them to. And what they found was that elite athletes realized a post-marathon increase in a very specific type of organism that is good at reducing lactate and generating propionate, Vianella atypica. And they also were able to see that at the gene level, all of the genes in a major pathway that do this conversion I'm referring to were upregulated fairly substantially. That did not occur over the same time course for the sedentary group that was not involved in the marathon, and they went on to demonstrate this proof of concept in animal models. So it's an interesting concept about how activity levels could influence microbiota in a way that actually makes it easier to exercise and get physical activity. And then the final set of proof of principles I want to talk about are from some of my own research, which tend to come from more large population-based research, where I'm not looking at extreme phenotypes, but I'm looking at the role of generally oral microbiota and to some degree gut microbiota and their influence on the early development of cardiometabolic disease. So we have about 1,100 people enrolled in a longitudinal cohort study, and we're wondering whether or not oral microbiota can predict insulin resistance, progression of fasting glucose, incident prediabetes, et cetera. And these first set of results, what we found is that a subset of about 20 organisms, pictured here, out of roughly 700 oral organisms, were very good predictors of longitudinal glucose change over a 2-year time period. We found that about half of them predict worsening glucose, but about half of them seem to be health associated, so higher levels meant better glucose outcomes over 2 years. We then took a ratio of the good to the bad, and we looked at that ratio in all individuals, and we found that those individuals with the microbial dysbiosis index in the highest third versus lowest third in the population had about a 6 milligram per deciliter greater increase in fasting glucose only over 2 years. Now, if you scale that up to 5 years or 10 years, these are actual risks that translate into conversion to prediabetes and diabetes. So it's another interesting and thought-provoking idea about the role of microbiota in cardiometabolic risk. And then we've gone on to look more specifically at particular mechanisms. So as it turns out, maybe 20% of the organisms in your mouth can actually convert dietary nitrates found in green leafy vegetables and beets into bioavailable nitric oxide. And that's important because, as many of you know, nitric oxide has for some time now been known to be quite important for vascular health and maintenance of good blood pressures and lowering of insulin resistance. So the idea is that you eat a green leafy vegetable, organisms in your mouth help convert a good portion of that to bioavailable nitric oxide, which helps keep blood pressure low and might mitigate against insulin resistance and fasting glucose. So we calculated a score of nitrate reducers, and we found that individuals in the highest tertile of nitrate reducers versus the lowest tertile of nitrate reducers had reduced levels of insulin resistance, they had reduced glucose levels, and they also had reduced blood pressure. And this is in a population-based setting, individuals that would be very reflective of what we'd probably find in a group like this. So we think that they potentially have meaning to larger population-based samples. Okay. So those are some what I think to be thought-provoking examples about the potential for the human microbiome. A lot more work needs to be done to really demonstrate this in large human populations for hard health outcomes. But if you're with me and you think that this is worth looking at, and there's a potential connection for prevention, we need to know a lot more about what actually induces and shapes the microbiome. Okay? And for the last few slides, I just want to introduce this concept of microbiome as it relates to green space. So we know that green space has been well associated in many studies with a number of health outcomes, and that's been discussed in several talks here today. The exact mechanisms underlying this are not exactly clear, and the prior hypotheses that were delving into this, such as the hygiene hypothesis, often tended to focus on a more narrow set of pathogen exposures, that actually lack of pathogen exposure early in life didn't train your immune system properly and you overreacted later in life, and it led to respiratory and allergic diseases. But these hypotheses generally haven't thought very much about the microbiome more broadly or cardiometabolic outcomes specifically. So an emerging idea building on and adding new dimensions to these old hypotheses is what's referred to as a microbiome rewilding hypothesis, which posits that ecological restoration of urban green spaces can rewild urban spaces to a habitat that is health promoting, and the underlying mechanism is believed to be potentially by increasing biodiversity and better and more exposures to health promoting microorganisms. Okay? So the concept is that this is kind of what we evolved from with more trees and less concrete. This is the state we're currently in with less biodiversity, and if we can convert back to something that's a little bit more intermediate, maybe we can bring back some of those earlier and beneficial microbial exposures. Okay. What I think is really neat and interesting about this hypothesis is that it really is adding a new dimension to and building on prior hypotheses that had some hint of microbial exposures in the green space area. And it's very interventional. So older hypotheses tended to focus on how we got here, whereas the newer hypotheses are focusing more on interventions that could actually be done in real urban settings to try and manipulate chronic disease health outcomes. So it holds a lot of potential in that space. And along with this, I should say that what people are thinking of are not just planting a few more trees and getting more grassy areas and larger parks, but it's a whole new and multidimensional approach termed microbiome-inspired green infrastructure, or MIGI, as I've pronounced it. It's not my construct, but that's my acronym for it. And as I said, it refers to multifunctional green spaces that would be designed to specifically increase more human interaction with the broader set of biodiversity and microorganisms that could be beneficial to health. So they're thinking of things like green roofs on every building and green walls to restrict or reduce noise rather than physical structures such as concrete and wood. Generational approaches that really think about where people are at and how they use space. It was a great point made in an earlier talk that we spend 90% of our time indoors. If we spend 90% of our time indoors, what relevance does green space have in the microbiome in that context? So we may have to think about how to get people outdoors and moving to those green spaces a bit more frequently. And I love the example I was reading about with Pokemon Go, which I'm sure is very passé by now, but my kids were very into it a few years ago when we lived in New York City, and we were all over the place trying to get to Pokemon Go stops. But you can imagine that for a good portion of the population, you can find ways to engage them in the activities that they like by getting them outdoors. I spend most of my time, as many of you do, behind a computer in an office with the door closed as an introvert. And if you could get more internet in green spaces, for example, and screens that work better in sunlight, I'd probably spend a little bit more of my time meeting outside. Another innovative idea in this space is foraging, the literal concept that on the way to work you might pick some nuts and berries and snack on them as you're walking through a park. That would be amazing. Not practical in the near term, but it's something that they're thinking about in this space. Okay. So there are a number of unanswered questions that we could talk about in the discussion. I just want to frame it a little bit and pose the idea here that we need to think about going forward. And we need a lot of research and data collection to inform. So, for example, is it the microbiome of the soil, of leaf surfaces, or is it the airborne microbiome that is the most important to real health outcomes, or is it some combination of all of these things that we really need to be thinking about? And to be honest, we really have absolutely no idea. We also don't know how these things really impact the microbiome. Can planting more trees and biodiversity and nuts and berries, et cetera, actually bring a microbiome that's diverse enough to have a human host impact? These are questions that remain to be answered. And what is the impact, of course, and the role of modern landscaping practices and pesticides and herbicides, et cetera, because usually those things tend to come along with more green space. If we're going to have green space, people generally want it to be green, and that can lead to other unanticipated consequences. There are a number of other questions that I posed here, and I believe you'll get the slides, so I'll let you read through those on your own, but needless to say, we really need to know a lot about what metrics of microbiome actually predict hard CVD outcomes, and we're a long way from getting that hard evidence. I want to end by saying one brief thing about unintended consequences, and using an example from my good friend and colleague, Gina Lavasi, who co-organized this event. I would imagine that there will be a number of very surprising unintended consequences if we move forward in this whole green space environment. And I'm talking about biologically unintended consequences, not just the socially unintended consequences like angry people who don't have a place to park their car in Barcelona. Once we get past that, there will certainly be biological consequences. So Gina and her team several years ago looked at tree canopy coverage in New York City, assuming that they would find that if you lived in tree canopy covered areas or areas with more tree canopy coverage, you'd have better health outcomes, respiratory health outcomes. And what they actually found is that people in those areas were more likely to have asthma and more likely to have various tree pollen allergies, which was remarkable. And there are various threats to validity with this study, as she's talked about at length, but they were very clever in their approach and careful and looked at it in many different ways and seemed to rule most of that out. And it seems that, at least in this place and context, that being closer to more tree coverage may have had some very real and unanticipated consequences for some of the very health outcomes we would hope to prevent. I'm not sure what happened in terms of cardiovascular disease, but in another 20 years, those data may be available. So I'm going to conclude simply by saying that we know that there's some potential connection between microbiome and cardiometabolic disease, but we don't really know what shapes the microbiome at all. And the urban green space environment is one potential big high-level exposure that could influence lots of people and have a real outcome if, in fact, the influences on the microbiota were meaningful. So I would make a pitch to city planners and chief epidemiologists to maybe start incorporating some of these metrics and measures into their environmental monitoring as we go forward. And I will stop there and leave you with this slide. Thank you.